Okay, so we are going to talk about genetics today, chapter 10 in your book. We're going to start with the patterns of genetic inheritance, and we're going to talk about Gregor Mendel and his laws of heredity. We're going to talk about some terminology and then how to do crosses. So Gregor Mendel was known as the father of heredity. He was an Austrian monk, and he was working in the 1860s. And he was actually working at the same time as Charles Darwin was on his theory of evolution and natural selection. So had they been able to communicate, who knows what we could have gotten done back then. The important thing is that his work is still repeatable today because he used the scientific method and he took detailed notes in all of his work. He was objective and he had a strong mathematical background, so this helped him out as well. You have to remember he's working in a time where he didn't have microscopes. They didn't know what cells really were. So this is revolutionary work. He worked with true breeding garden peas. And what true breeding means is that we all have two alleles, one donated from one parent, one from the other. True breeding means those alleles are the same. And we'll talk more about terminology later. So we use these garden peas because, first of all, they were very easy to grow. They can selectively cross-pollinate, so he could pick and choose what he wanted to cross-pollinate. And they have a very short generation time, meaning that he could study generations in a very short period of time. So his particulate theory of inheritance replaced the popular blending theory. The blending theory back then basically thought that the offspring were an intermediate of the two parents. So their two parents kind of blended together to form this intermediate phenotype. So, for example, if you crossed red flowers with white flowers, you would get pink flowers. And then if you crossed pink flowers with pink flowers, you would still get pink flowers. So with the red and white makes pink, that's an intermediate phenotype of the parents. But pink times pink sometimes equals red and white flowers. So this directly contradicts the blending theory, because the blending theory would not see those original parental phenotypes come back. So if it was blending, true blending, pink times pink would always equal pink. But what Mendel found was that the red and white flowers also came back in that generation. So the law of segregation basically states that individuals have two genes for each trait. So individuals are diploid. So if you look at the picture on the right-hand side, you have the diploid individuals down there at the bottom, the man and the woman. These genes then separate during the formation of gametes. So chromosomes separate during meiosis, during anaphase 1. Gametes only have one factor or gene for that particular trait because, as a result of meiosis, the gametes are haploid. So if you follow that picture around, you go through meiosis and you form those haploid gametes, the sperm and the egg. Fertilization then restores the diploid number because the two genes for each trait combine. So you have the egg and the sperm combine their chromosomes. So now they restore the diploid condition and they form a zygote. That zygote will then go through mitosis to develop back into an adult. So you have this constant life cycle where the diploid adults are creating haploid gametes through meiosis, and then the egg and the sperm fertilize, creating the diploid zygote, which then goes through mitosis to become an adult. He also had the law of dominance. So of these two genes, one codes for a particular trait that's going to be expressed as long as it is present, and the other one is going to be unexpressed. So the one that's always expressed when present is called the dominant allele, and the one that is unexpressed unless both genes are, both factors, both alleles are recessive, is called the recessive allele. So if the dominant allele is present, that trait will be expressed. they mask the expression of the recessive genes. But the recessive trait will be expressed if there isn't a dominant gene present. So remember, we have two alleles. Everybody gets two genes, one from mom, one from dad. So we have flower colors. Purple is dominant, white is recessive. So purple will always be seen as long as that purple allele is present. So whether there's two purple alleles or whether there is one purple allele and one white allele, those flowers will be purple. White flowers come from the presence of two white alleles, so no purple at all. So the genes, if we use uppercase P and lowercase p, the genes that a purple flower can have are two uppercase P's or an uppercase P and a lowercase p. The genes that a white flower plant can have are only two lowercase p's, because the recessive trait can only be expressed if both alleles are recessive. So the law of independent assortment is another law that he had. Basically, it says that each pair of alleles assorts independently of the others during gamete formation. So during metaphase 1 of meiosis, each homologous pair is going to align randomly at the equator. So all possible combination of genes can occur in a gamete. So what that means is that it's not going to be one parent alleles are on the top, one parents are on the bottom. It doesn't mean that all the dominants are on top, all the recessives are on the bottom. It means that they align randomly. So it could be, one parent has two chromosomes in a row, and then the next parent has two or three chromosomes in a row. Or it could be there's a few dominant traits in a row and then recessive. It's all completely random. So if you look at the example on the bottom there, you have the dominant alleles on the top and the recessive alleles on the bottom in the first picture. So the possible gametes that can be formed are big A, big B, and little a, little b. Because remember, anaphase is going to pull them apart. So the dominant genes will be pulled one way, the recessive will be pulled another way. Or they can line up where it's the dominant A, but the recessive B on top, and the recessive A and the dominant B on bottom. So now the gametes that can be formed are big A, little b, or little a, big B. Because remember, again, anaphase is going to pull those apart. So the big A, little b will go one way, the little a, big B will go the other way. And those are the gametes that will be formed. So gametes are not genetically identical. They have a variety of gene combinations, which means that meiosis leads to genetic, hopefully you said variability. So inheritance of eye color, for example, is independent of freckles because these genes are not on the same chromosome. So someone can have blue eyes with freckles and someone can have brown eyes with no freckles. If they mate, the children can end up with blue eyes and no freckles and brown eyes and freckles because they are not on the same chromosomes. So they're going to align separately, independently of each other, and they're going to form gametes. So you can have completely new combinations. So, terminology. The P generation is the parental generation. This is the original parental line. It is purebred or true breeding, meaning that the alleles are the same. So two uppercase P's or two lowercase p's. The F1 generation is when you cross the dominant with the recessive. Dominant true breeding with recessive true breeding. So you cross big P times little p. The offspring produced by this generation, the F1 generation, are going to be hybrids or heterozygous. They're going to be big P, little p. Then if you cross that generation, so you cross big P, little p times big P, little p, now you've got the F2 generation. The phenotypes are going to be 3 dominant to 1 recessive. So 3 purple to 1 white. And F stands for filial, by the way. So Mendel looked at flower color, seed shape, pod shape, flower position, and plant height. Flower color dominant, purple. Seed color is yellow. Seed shape is round. Pod color is green. Pod shape is round. Flower position is axial. Plant height is tall. The other side are the recessives. So again, he crossed pure breeding plants. So it was big A times little a. So the hybrid F1 generation all came out the dominant trait. And when you cross that F1 generation, the F2 generation basically came out with a 3-to-1 ratio. Now keep in mind, it's not going to be exactly 3-to-1, but for the most part, they all round to 3-to-1 as a ratio. So you end up with 3 dominant phenotypes, 1 recessive phenotype. So we have 3 purple, 1 white flower, 3 yellow, 1 green seed color, and so on. So some terminology. A gene is a unit of heredity. The DNA, it's a segment of DNA, and these are found on chromosomes. The locus is the specific location where that gene is located. So if you look at the picture down there of the homologs, those are your chromosomes. So the alleles are variations of genes. The locus is just where they're at on that particular chromosome. So the gene is the trait. The allele is the alternative form of those genes. So going back to flower color, the gene is for flower color. The alleles are purple and white. So you have the gene is the trait, the alleles are the variations of that trait. Okay, so the flower color is the gene, purple and white are the alleles. So some concepts that are important to understand. It takes 2 genes to express a trait, 1 from each parent. Multiple genes are found on a chromosome. In man, we used to think that we had about 100,000 genes because we used to say, well, we're the smartest, most complex creatures on Earth. We have to have a lot of genes. What we found with the Human Genome Project, though, is that we still do not exactly know how many we have, but we know it's a lot lower than that 100,000. We know it's closer to about 23,000, 24,000 genes. But still, let's say we have 23,000 genes. We only have 23 pairs of chromosomes. And again, each set of chromosomes is going to have the same genes. So 23,000 genes on 23 chromosomes, so you can do the math there. So each chromosomal pair is going to carry specific traits. And that was part of what the Human Genome Project was, to map our chromosomes and figure out which traits were carried on which chromosomes. So here we have chromosome 1, chromosome 22, and then X and Y. Chromosome 1, 246 million base pairs. Chromosome 22, 49 million base pairs. The Y chromosome, 50 million base pairs. The X chromosome, 153 million base pairs. So you can see how many different genes are on these particular chromosomes and size difference with these chromosomes. So which 2 are autosomes? Hopefully you said 1 and 22. Sorry, I gave you too long there probably. But the X and the Y, the 23rd pair, are the sex chromosomes. So, again, dominant versus recessive. The dominant gene is the gene that will be expressed as long as it's present. It masks the expression of the recessive allele, and it's denoted by capital letters, so uppercase T. Recessive is the gene that will only be expressed when the dominant gene is not present. So this is the gene that's masked by the dominant allele, and it's denoted by a small letter, a lowercase t, for example. Now, when you're figuring out what letters to use, you usually use the first letter of the dominant trait. So for the flowers we were talking about, it was P because purple is the dominant trait. For tall versus short, you would use T. Okay? So, alleles, they can be the same or they can be different. If they are the same, that says the offspring is homozygous. So uppercase T, or lowercase t. So this was Mendel's P generation, also called purebred. Heterozygous means that the alleles are different, so big T, little t. This was Mendel's F1 generation, and they're also called hybrids. So you can have a homozygous dominant, a homozygous recessive, or a heterozygote. So the homozygous dominant would be big T, big T. The homozygous recessive would be little t, and the heterozygous would be big T, little t. Genotype refers to the genetic makeup of an offspring, so the particular genes that the offspring has. So again, homozygous dominant is big T, homozygous recessive is little t, and heterozygous is big T, little t. Phenotype refers to the physical appearance of an offspring, so remember phenophysical. We'll start with the PH. Genotype, genes. So the physical appearance, we're talking if it's tall versus dwarf, if it's purple or white. So if it is uppercase T, or uppercase T, lowercase t, that means it's tall. So if it's homozygous dominant or heterozygous, it's going to be tall. However, if it's homozygous recessive, little t, then it's a dwarf plant. So tall and dwarf are the phenotypes. Big T, little t, big T, little t are the genotypes. So, looking at these alleles, the blue eyes is recessive, and that's blue. Brown eyes is dominant, and that line is brown. So, individual A, what is the genotype? So, if you look at it, look at your key. Uppercase B is brown, lowercase b is blue. So that individual is going to be big B, little b. What is the phenotype for that individual? Brown is dominant, so it's going to be brown. Individual B is homozygous. So what does that tell you? There's two brown alleles. So the genotype would be big B. The phenotype would be brown eyes again. Now individual C is homozygous recessive. So you have two blue alleles. So the genotype would be little b, and the phenotype would be blue eyes. Okay, so monohybrid crosses look at one single trait. So it takes two genes to express a trait, remember. One from one parent, one from the other parent. Genetic problems are a way to statistically predict what the offspring's genotype and phenotypes may be. It involves probabilities, and the more offspring that you have, the closer you can get to the prediction. So what we use are called Punnett squares to do genetic problems and offspring predictions. So remember, two alleles, two genes, so we have two gametes that can be formed for the egg, two for the sperm. So you put those across the top and along the side. Now it doesn't necessarily matter if you always have the eggs across the top and always have the sperm across the bottom. That doesn't really matter so much. What matters is that you keep everything in line, and I'll tell you why. So the little n, those are the alleles donated by the gametes, so they're haploid again, formed by meiosis. And the offspring are then going to be 2n because they are diploid. it's going to represent fertilization. So if that little first sperm there fertilizes that first egg, that restores a diploid. If the first sperm fertilizes the second egg, it restores a diploid. If the second sperm fertilizes the first egg, it restores a diploid, and so on. So, using Mendel's law of segregation here, if the parent genotype is big T, little t, then the possible gametes that can be formed are going to be big T or little t. These are formed during meiosis, remember. And remember, the gametes are formed, so one allele goes to one, the other allele goes to the other. So if you're going to cross big T times big T little t, remember uppercase T is short, uppercase T is tall, lowercase t is short. So here we're going to combine, or cross I should say, two heterozygote parents. Heterozygote meaning big T little t. And then you simply just match up the boxes. So your first box you got big T. Your second box you got big T little t. Sorry. Your third box you have big T little t. And your fourth box there you have little t. So you're going to end up with 3 dominant tall phenotypes and that 1 recessive short phenotype. So it's that 3-to-1 ratio again. So what is the chance of producing a recessive offspring? So you have to look at your little Punnett square and look at the chance of producing that recessive offspring. Hopefully you got 1/4 or 25%. So what is the chance of producing 2 recessive offspring? Now you have to think. Genetics doesn't have memory. So it's not like if you have 4 children, those are the 4 children you're going to have. You're not necessarily going to have, these plants here, we'll say. If you cross these 2 plants 4 times, they're not necessarily going to have 1 homozygous dominant, 2 heterozygous, and 1 recessive offspring. That doesn't happen. So you have to keep thinking. What are the odds that you flip a coin heads or tails? Hopefully you said 50%. So let's say you flip a coin and you get heads. Now what are the odds that you flip it again and get heads? Well, hopefully you said 50% still, because it restarts every time, okay? Genetics doesn't have memory, as I said. So every time you flip that coin, you still have the same probability. The probability of having a boy versus a girl is 50%. If you have 1 child, the probability of having another boy or girl, still 50%. But this is asking, what's the chance of producing 2 recessive offspring? So that's like saying, what are the chances of having 2 girls in a row or 2 boys in a row? So for this case, you have to think, okay, it's 1/4 to have 1, multiply it by 1/4 to have 2. So it's going to be 1/16. Okay? And the reason that you're multiplying them is because you're looking at the possibility of 2 events happening. So you don't want to know what's the chance that you have a recessive for this time and then a recessive for next time. You want to know what are the odds that you'll have 2 recessive. So that means you have to multiply. So just like what are the odds that you have 2 girls in a row, 2 boys in a row kind of thing. So you have to multiply those odds. So the law of probability can be expressed in 3 different ways. A ratio, which is just the number of dominant to the number of recessive. So 3-to-1 in our last example. A fraction, which is just the number with that specific trait over the total. So 3/4 dominant, 1/4 recessive. Percentages, you divide the numerator by the denominator and multiply it by 100. So if you just think about fractions and percentages with counting money, 1/4 is 25%. That's a quarter. 2/4 is 50%. that's 50 cents. 3/4 is 75%, so that's 75 cents. And 4/4 is 1 whole, so you're up to that dollar now. It's 100%. As I said, probabilities don't change with each offspring because heredity does not have memory. Probabilities are only predictions, they're not reality. So just because you, like I said, have 4 offspring, it doesn't mean they're going to each be that particular one in the Punnett square. But the more offspring you have, the closer reality comes to the statistics. So 19 kids and counting, for example. If you look at the chart over there, they had a boy and then a girl and then a boy, but then they had 3 girls in a row. Then they had 2 boys. Then they had another girl, but then they had 6 boys in a row. But if you count overall, they have 10 boys and 9 girls. So that's really close to that 50-50. So if Michelle Duggar has another child, what is her chance of having a boy? Hopefully you said 50% because, again, it doesn't change. It's individual for each time. If she has another child, what is her chance of having a girl? Hopefully you said 50%. So, how do we find out the genotype of a dominant phenotype? Think about this. Dominant phenotypes, so we'll say tall. They can be big T, or big T, little t. We look at them and we see tall, but we do not know the genotype. However, short is little t, and we know that. So if we see a short plant, we know that it's little t. So in order to determine that genotype of that dominant phenotype, you mate the individual with the unknown genotype with the recessive trait. So you mate the big T, or the big T, little t. You mate the tall individual with a short recessive individual. And then you look at the offspring. So if you have a dominant phenotype and mate it with the recessive, and you come up with all tall offspring, that means that the dominant phenotype has to be big T, because that's the only thing he can contribute is the big T. The only thing the recessive can contribute is the little t. So if they all come up tall, you have a homozygous dominant phenotype. However, if you look at the offspring and half are short and half are tall, that tells you that the dominant phenotype had to be a heterozygote, because that's the only way you'll get half tall and half short. Okay? So if you don't have any recessive offspring produced, chances are it's a dominant homozygous, so big T. If you have recessive offspring, that means that both parents have to carry that recessive trait. So they have to have at least one recessive allele. So that makes them heterozygous, big T, little t. Okay, dihybrid crosses. Di means 2. So now you're looking at 2 different traits located at 2 different points. They're non-linked, different chromosomes. So the genes for Drosophilus, wing type and body color, for example, they're on different chromosomes. So we look at wing length and body color. Now, if they were linked, it gets more complicated, and that's for an actual genetics class, so don't worry. So wing length, long is dominant, short is recessive. Body color, gray is dominant and black is recessive. So the genotype for a long-winged black body. Now the long wings, long is dominant. So we do not know if it's going to be big L or big L, little l. But black body we know is going to be little g, because it's a recessive. Short-winged gray body. Now again, short is recessive, so we know that has to be little l. but gray is dominant. So we know it's big G, but then we don't know what the other one is. For a long-winged gray body, now we don't know either actual genotype. So we know there has to be a big L and a big G, but just the simple presence of that big L and big G makes it dominant. Short-winged black body, however, we know both because that's both recessive traits. So it's 2 little L's and 2 little G's. So think about this. It's just like if we looked at long and we looked at gray. You have the gametes for the long, the gametes for the gray. Okay? So 4 letters across. So you have 2 traits now. So for one trait, you had 2 letters. So for 2 traits, you're going to have 4. So how would you determine the genotype of a long-winged fruit fly with a gray body? Hopefully you said do a test cross. What would you mate your unknown to? Double recessive. So little l, little g, little l, little l, little g, little g. So the short-winged fruit fly with the black body. You're going to look for recessive offspring with one or both recessive traits, and that will help you figure out the parental. So inheritance of wing length and body color are independent of each other. So if the genotype of an individual is big L, big G, little g, then what possible gametes are going to be formed? Okay, big L, little, or big L, big G. So you have the first 2 there. Then you have big L, little g. Then you're going to have little l, big G, and little l, little g. What you want to do is do the first with the third, so big L, big G. Then you want to do the first with the fourth, big L, little g. Then you want to do the second with the third, little l, big G, and then the second with the fourth, which is little l, little g. So, just like that. So if we cross big L, big G, little g, so 2 heterozygotes, what are the possible gametes we have? So we have big L, big G, big L, little g, little l, big G, little l, little g. For both. They're both heterozygotes. Now it's just like a regular Punnett square, except this is more of a rectangle. You just match up the boxes. It's very important to make sure that you keep the traits together. So you're going to keep the L's together and you're going to keep the G's together. When you're writing, the dominant always goes first if there's one present. So you're going to end up with these 16 genotypes. When we're doing dihybrid crosses, as you can see, the genotypes are kind of spread all over the place. So we don't care about genotypes as much as we care about phenotype here. So the children, the offspring here of these individuals are going to have a 9:3 to 3:1 ratio for phenotype. So the physical reality, the physical appearance. So we're going to have long gray, which are the 2 dominant, 9 of them, and there are the 9 circled. Long black, 1 dominant, 1 recessive. Those are in yellow, so we have 3 of them. Short gray, which is the other recessive and dominant. So there those are, again, 3. And then 1 little double recessive at the end there, short black. So you're always going to have this phenotype. So if you're ever asked, if you do a dihybrid cross of 2 heterozygotes, what is the phenotypic ratio of the offspring? It's always going to be 9 to 3 to 3 to 1. The 9 are both dominant traits. The 2 3s are going to be one dominant, one recessive, and then the other dominant and the other recessive. And then the 1 is going to be the double recessive. Okay? Now, if we do a heterozygote times a double recessive, so big L, big G times little l, little g. The gametes that you can get from the heterozygote don't change. Big L, big G, big L, little l, big G, little l, little g. But the gametes for the double recessive do change because all the double recessive can give is little l, little g. So that's all they can give. So you have the same gamete for all 4. Same thing though, you cross them. Now, what do you do from here because it's the same? Well, you simply draw arrows because it's going to be the same down each one. You're going to have big L, big G for all those first 4 boxes. Big L, 2 little g's for those second boxes, and so on. So the ratio we come up with here is 1 to 1 to 1 to 1. So if you're ever asked if you cross a dihybrid cross, a heterozygote with a pure breeding, it's going to be 1 to 1 to 1 to 1 phenotypic ratio. So P, parental, pure breeding, it's going to be big L times little l, or big G times little g for the monohybrids. The F1 offspring generation, it's going to produce all hybrids. So big L, or big G. And the phenotypic ratio for the offspring are going to be 3 to 1, always. Now for a dihybrid cross, the P generation, again, a double homozygous dominant with a double homozygous recessive. Dihybrid is 2. The F1 offspring are going to be all heterozygotes, double heterozygotes, so big L, big G. And the F2 offspring, when you cross those hybrids, you get 9 to 3 to 3 to 1. So, parent cross, big T times little t. The offspring phenotypes, they're all going to express the dominant trait because they're all going to be big T, little t. Now, what if we cross big T times big T? What are we going to get? Three to 1. 3 dominant to 1 recessive. If we cross big T times little t, what are we going to get? One to 1. 1 dominant, 1 recessive. Now, the thing is, if you ever doubt your answer or you just don't know the answer, do a Punnett square real quick. Just, write out big T across the top, little t on the bottom. Do your Punnett square. You're always going to know that. So if we do dihybrids now, pure breeding, double homozygous dominant, double homozygous recessive, what are we going to get? They're all going to express both dominant alleles because all of the offspring are going to be heterozygous for both alleles. Now, if we cross 2 heterozygotes, what's the offspring phenotype? Nine to 3 to 3 to 1. And if we cross a heterozygote for both times the double recessive, what are we going to get? One to 1 to 1 to 1. And again, just like with the monohybrids, if you're in doubt, do the Punnett square, or rectangle, I should say, for dihybrids.